Hello everyone, myself Manasi Rajan Korgaonkar. I am going to explain this poster based on enzyme urease. So in this poster we are going to learn about history, reaction, structure, mechanism, production and various applications of enzyme urease. Before moving towards explanation, first let us get familiar with the term enzyme. So what are enzymes? Enzymes are basically biocatalysts, which means these enzymes are from biological sources, which are high molecular weight proteinous compounds that catalyze or speed up biochemical reactions. So now I am going to explain about one such enzyme, that is enzyme urease. So as you can see in this poster, urease has an enzyme commission number, that is EC 3.5.1.5, which tells us that it belongs to the class hydrolases and superfamily of amidohydrolases and phosphotieesterases. It acts on carbon-nitrogen bonds other than peptide bonds. It is a nickel-containing metalloenzyme of higher molecular weight, that is 480 kilodalton or 545 kilodalton. So now let us move further to the history of the enzyme urease. So as you can see, this scientist, Frederick Alphonse Musculus and James B. Sumner. So these were the scientists who were responsible for identification of enzyme urease. Urease activity was first identified in 1876 by Frederick Alphonse Musculus as a soluble ferment. In 1926, James B. Sumner showed that urease is a protein by examining its crystallized form. Sumner's work was the first demonstration that a protein can function as an enzyme and led eventually to the recognition that most enzymes are in fact proteins. Urease was the first enzyme crystallized. For this work, Sumner was awarded with the Nobel Prize in Chemistry in 1946. The crystal structure of urease was first solved by P.A. Karplus in 1995. So the image which we can see here is the 3D model of urease from Klebsiella aerogenes, which was first solved by P.A. Karplus in 1995. Now let us see the reaction part of enzyme urease. So reaction has a substrate and a product. A substrate is a molecule that an enzyme reacts or binds with to catalyze any chemical reaction. And these substrates is transformed into one or more products. So now let us see what are the substrate and products in this reaction. So this reaction involves hydrolysis of urea, which is the substrate, by the enzyme urease, which leads to the production of the two products, that is, one is the ammonia and second is the carbon dioxide. Now let us move further to the structure and mechanism part of enzyme urease. So the structure which we can see here is the schematic depiction of structure of active site of Bacillus pastöri urease. The active site contains two nickel ions with an interatomic distance of about 3.5 angstrom. The ions are bridged by a carbamylated lysine and an oxygen donor. In addition to the bridges, one of the nickel ion is coordinated by two histidines and a water molecule. The coordination of nickel 2 is similar to the one of nickel 1 and includes two histidines residues of water molecules and a terminally bound aspirate, which we can see here. Now let's see the mechanism part of enzyme urease. A number of proposals have been made regarding possible reaction mechanisms. One mechanism for the catalysis of the reaction by urease was proposed by Blakely and Journer, which includes nucleophilic attack by carbonyl oxygen of the urea molecule onto the 5-coordinate nickel. The second was the mechanism proposed by Hosinger and Karplus, which attempts to revise some of the issues apparent in the Blakely and Journal mechanism and focuses on the positions of the side chains making up the urea binding pocket. The third was the Kayerle and Magny mechanism. The mechanism proposed by Kayerle and Magny, which is one of the more recent and currently expressed views of mechanism of urease, and is primarily based on different roles of the two nickel ions in the active site. The main structural difference between the Cairley-Magny mechanism and the other two, that is Blakely journal and Hosinger-Karplus mechanism, is that it incorporates a nitrogen oxygen bridging urea that is attached by a bridging hydroxides. Now let's see how urease enzyme is produced. So urease is an enzyme produced by ureolytic microorganisms like Bacillus pasteurii, which hydrolyzes urea into ammonia and carbon dioxide. Urease is found in large quantities in jack beans, soya beans, and other plant seeds. It also occurs in some animal tissues and intestinal microorganisms. So now let's see the applications of urease enzymes. So microbial urease has wide applications in fields like biotechnology, agriculture, medicine, as we can see these three pictures. So urease is also being studied in clinical samples, and it is related to the virulence of pathogenic microorganisms, contributing to urinary stones, gastric ulceration, etc. In last 2 decades, urease has also switched from clinical relevance to geotechnical engineering and applied biotechnology. So let us see the industrial applications of urease. Urease has many industrial applications in food industry as well as medical industry, which includes diagnostic kits for measuring urea in alcoholic beverages, in biosensors of hemodialysis, systems of measuring blood urea as a probe for detection of heavy metal ions in food sample, groundwater, industrial effluents, and environmental monitoring. So I hope this poster presentation has helped you to learn about enzyme urease. Thank you.